Carlos Robello. Carlos obtained his PhD at the University of Granada in Spain, more specifically at the Instituto López Neira de Parasitología y Medicina, belonging to CSIC. And he has been working on molecular biology and pharmacology of trypanosomatids. Currently, he's based at the University of La República and the Pasteur Institute at Montevideo, Uruguay, where he heads as well, a very productive research group and as well a platform of deep sequencing and microarrays. His work is mostly focused on the molecular biology and genomics of trypanosomatids and their interaction with host cells. In particular, his group has characterized virulence factors in Trypanosoma cruzi, the causative agent of Chagas disease. These factors are of relevance in the infection and the persistence of the parasite. And in the last years, he started to work on the genomic characterization of T. cruzi strains and on the transcriptional changes occurring during the infection, both in parasites and the host cells. So, Carlos, thank you very much. And the audience is yours.
Okay, can you hear me? Yes?
Yes.
Okay, thank you very much, Ariel and Marie-France, for the invitation, for the presentation, and congratulations, because I think this meeting is really fantastic. This approach between metabolism and mathematical models. So thank you very much. I'm very happy to participate here. I will talk about T. cruzi, Trypanosoma cruzi, the agent of Chagas disease. Just to mention that this is the cycle of the parasite, where it is a vector-borne disease, Chagas disease, and the parasite is unicellular, is a protozoan parasite, Trypanosoma cruzi, and has several stages during its life cycle. Okay, this is this is the epimastigote stage, that is the insect stage. The trypomastigote is stage that is infective. Epimastigotes are proliferative, whereas trypomastigotes are not proliferative. Is the infective form of the parasite. They will infect cells in mammals and will transform to amastigotes. And after that, during several hours or days, they will be back trypomastigotes. We name these cell-derived trypomastigotes, yes, that are also non-replicative. Sorry, I say proliferative. I should say replicative form of the parasite. Okay, so I will talk about this interaction between the parasites and the host cells, because during the mammal part of the cycle, they are intracellular, whereas in the insect, they are not intracellular, okay? So this is our reference, the epimastigotes, and I will talk about this, also these stages, amastigotes and cell-derived trypomastigotes. One important point in the biology of T. cruzi is that they can invade almost any kind of cell. That is really impressive, because in the case of other similar parasites, like Leishmania, for example, they infect macrophages, phagocytic, professional phagocytic cells. In the case, for example, of African trypanosomiasis, Trypanosoma gambiense, rhodesiense, and brucei, they do not infect cells. In the case of T. cruzi, they are able to invade fibroblasts, macrophages, epithelial cells, adipocytes, cardiomyocytes, etc., etc., etc. So this impressive ability of T. cruzi is related to several proteins, most of them surface proteins that are in T. cruzi, but also intracellular proteins that participate in the process of adhesion, invasion, and infection. And there are several receptors in the host cells that have been related to infection. but this schematic view of the infection of T. cruzi just to see different ways to infection, but we don't know. I mean, in each cell type, there are some receptors that are relevant and other receptors that are relevant in other cells, and also the surface proteins or the intracellular proteins that participate in the infection are different depending on the kind of cells, taking into account that they can invade those different cell types. The invasion process also is different depending on the cells. It can be related to acting to the lysosome recruitment. Acting can participate actively in the invasion, and also PIP3, yes, but always they will invade, and they will be inside parasitophorous vacuole, yes. They will infect as trypomastigotes. After that, they will transform to amastigotes. They will replicate intracellularly, and after that, they will transform again to trypomastigotes, and they will be released from the cell that they infect. We name early infection this part of the cell cycle where in the first 24 hours we named the early infection, and what we wanted to see is what happens during this initial step of infection at the level of gene expression. So by interrogating the response of different cell types to infection, we intend to approach the molecular basis of this interaction and to know which factors of the host are necessary for infection and persistence. These factors can lead to new molecular targets and thinking host-targeted therapies, or combined therapies using trypanocidal drugs and host-targeted therapies. So I will show some results involving epithelial cells, cardiomyocytes, and macrophages, and the analysis of transcriptomics pathways, transcriptomic results in the early response to T. cruzi infection. This is our experimental design: human cells plus this strain that is DM28C. The relation, the MOI is 1 to 10, and we analyze the different in different times of infection. We extract RNA, we made transcriptomic, and we analyze what is happening. Okay, so in epithelial cells, in cardiomyocytes, and in macrophages. Okay, I will show. We have also done experiments in another models, but I will focus. The presentation is in these three types of cells. Okay, three types of cells. When we did the experiments in epithelial cells, what we found was that drastic gene expression changes were seen in the same first hours of infection. Most of the changes were in overexpression. Yes, what I am showing here is number of genes, and expression in red the overexpressed genes, in green the down-regulated genes. Okay, and there are several pathways that were affected. In particular, in this epithelial, these are HeLa cells. We found that most of the pathways were related to immune response. Yes, cytokine stimulus, neutrophil chemotaxis, inflammatory response. So there were several changes related with immunity, another related to apoptosis, and also genes related to a DNA damage response. Most of the upregulated genes remain in this condition during the early response to T. cruzi, whereas in the down-regulated genes are most of them different in time 0, 3, or 6 hours after infection. Okay, when we analyze the temporal regulation of all genes different pathways, we found that cholesterol metabolism related genes are affected, fatty acid metabolism also are affected, in particular elongases, and also the phospholipid metabolism. Some remarks about this response is that cholesterol metabolism, concerning cholesterol metabolism, the most remarkable changes occur in cholesterol transport related genes. Low density lipoprotein receptor, the oxidized low density lipoprotein receptor also were affected. In fatty acid, several genes for fatty acid metabolism, in particular members of the acyl-CoA synthetase family and elongases also, like this L7. And finally, the arachidonic acid and prostaglandin metabolism related genes were affected. Of course, we cannot conclude about metabolism with RNA-seq data, but this data give us an overview about what is happening, and we need to go to validation, to functional validation. I mean, we don't think that we are saying if we find differences in metabolism related genes, we do not conclude, okay, the metabolism is being affected, but we get some clues to go to the metabolic experiments. In this case, in the case of HeLa cells, we found that, for example, the lipid bodies were increased in the 6 hours after infection. The cyclooxygenase 2 gene was overexpressed also in response to the infection. And this is the oxidized receptor of LDL that also was overexpressed in response to T-cruzi infection. Also, we found IL-8 that was regulated by several, that is overexpressed, and several genes related to the DNA damage response. Okay, this is some results about IL-8 secretion, the NF-kappa B translocation to the nucleus immediately after infection. This is NF-kappa B. Here you can see that it's translocated in the nucleus 10 minutes after the infection and come back to the cytosol after 30 minutes. And probably it is one of the main responsible for the changes that we see during the infection of HeLa cells. In conclusion, concerning epithelial cells, lipid metabolism is severely affected. We found factors related to chemotaxis, in particular IL-8, that is related to chemotaxis of polymorphonuclear cells and angiogenesis, DDR-related genes, apoptosis, and another group of long non-coding RNA. That was what we found when used epithelial cells, HeLa cells, and when we infected them and analyzed the responses. When we went to human primary cardiomyocytes, we did the same experiment with the same strain, and also we have found a similar pattern in the sense that most of the changes were genes that were upregulated. Okay, these are, here we analyzed the first 12 hours of the infection of human primary cardiomyocytes. Most of the genes were upregulated, and some of them were downregulated. Here we show the control and the infected cells at different times, where green means downregulation and red means upregulation or overexpression. And when we analyze the different pathways that were affected at the transcriptional level, we have found a completely different pattern in cardiomyocytes. We found that cytoplasmic ribosomal proteins were one of the groups of the most affected genes in cardiomyocytes, and also genes related to oxidative phosphorylation, electron transport change, glycolysis, and Krebs cycle. These are examples of genes that belongs to oxidative phosphorylation, electron transport change, and cytoplasmic ribosomal proteins, where you can see that the change is drastic immediately after infection. We incubate the parasites for 1 hour. After that, this is the time 0, and each of these files represent one gene. So the idea here is to show that the change are really strong immediately after the interaction with the parasites. The affected genes are, for example, malate dehydrogenase related with metabolism, and genes related to complexes 1, 3, and 5 from the respiratory chain. And the changes are really high. For example, here are some subunits of the malate dehydrogenase have 10-fold changes. For example, some units of the ATP synthase 40 times, they change 40 times. They are 40 times upregulated, etc. So this gave us an overview of what was happening. So we went to analyze if, in fact, the respiratory chain was affected at the phenotypic level or functional level. We analyzed the oxygen consumption by using these different inhibitors of the respiratory chain, and we saw that 6 hours after infection, no significant changes were seen, but 24 hours after infection, yes, we have seen differences between the control and the infected cardiomyocyte. Here, we show the basal and maximal respiration, and this is spare capacity, and cruzi, after 24 hours of infection, change the basal, the maximal respiration, and also the spare capacity in cardiomyocytes. The difference between the changes that we see in the times in transcriptomic studies and in these functional studies is because it's probably because of the time between the transcription, traduction, and the creation of the complex. I mean, these proteins must be to the mitochondria and might be might participate in the different complexes. So that is why we think that we don't see change at 6 hours at the functional level, but 24 hours after that, there are important changes. So respiration is affected in response to the infection of cruzi. That is a point that is for us very interesting, is that when we did the same experiment in HeLa cells, no significant difference were found, meaning that this response is not a feature of cruzi in general, but is a specific response to cruzi in cardiomyocytes, only in cardiomyocytes, or at least in cardiomyocytes, not in HeLa cells. Also, we know in macrophages this does not happen in our experiments, and in fibroblasts also this does not happen. Okay, so this is a response related to cardiomyocytes. Since we found that there were changes in the spare capacity, we asked about biogenesis, mitochondrial biogenesis, and in fact, this is a specific genes and specific mitochondrial gene, and this is a nuclear gene. These are infected DNA from infected cells and DNA from control cells, and what we can see here is that there is an increase in the number of mitochondrial DNA, what suggests the biogenesis induced by T. cruzi infection. And here, when we use an antibody against cytochrome C in the infected cell, we can see the increase in the number of mitochondria related with non-infected cells. And also, we have found that there were changes in the shape of mitochondria, and we are now focusing in these changes and how in this model T. cruzi can induce changes at the fusion or fission in mitochondria. So we are starting a project in this area. So with these results, we asked about, we went to see if the mTORC1 pathway was involved in these changes, because mTORC1 through AKT can affect mRNA translation. And we have found several proteins, ribosomal proteins, overexpressed in cardiomyocytes, and also because it can favor the mitochondrial biogenesis, OCFOS, and Krebs cycles. So we went to analyze the mTORC1 response after T. cruzi infection, and in fact, we have found that AKT mTORC1 pathways are activated. These are several analyses where, for example, AKT, this is the protein AKT, and this is the phosphorylated AKT, 24 hours after infection, mTOR also, and several members of the pathway. All of them were activated. So this activation was responsible for the changes at the level of respiration, for example, in T. cruzi. When we added rapamycin, we could reverse the result. I will only show that if we add rapamycin, then we don't see differences between infected and uninfected cells in basal respiration, in maximal respiration, and we also here, when we see the mitochondrial DNA relative quantification, we see that with rapamycin, the relative quantification of infected cells is the same than the control cells. Okay, so this confirms that is the mTOR pathway that is activating these changes. So we have found that in cardiomyocytes, energy metabolism and protein synthesis are the most affected pathways at the level of gene expression, and there is a correlation with the phenotypic, the function of these cells. So we have found basal and maximal respiratory rates that were different. Increasing respiratory chain generate growth that can explain the donut shape of mitochondria, and this was related to mTORC through AKT activation. So this is what we found in cardiomyocytes. So the results with cardiomyocytes were completely different than those results obtained with HeLa cells. And finally, just I will show only one or two slides about this. In macrophages, when we use THP1 macrophages, that are human macrophages, we have found drastic differences at the level of gene expression, also at the level of splicing patterns of profiles, and this is a representative heat map that shows that in macrophages, change drastically. These are the non-infected cells. These are two replicates, and this is two replicates of infected cells, and they have inverse patterns of gene expression, also at the level of isoforms. Just show that in macrophages, there are drastic changes, not only at the gene expression level, but also T-cruzi alters alternative splicing patterns, and the most affected pathways were apoptosis, immune response, autoimmune, and infectious diseases. So in the general conclusion is that T-cruzi induces cellular reprogramming through changes in gene expression patterns. Each cell type has different responses, and this phenomena is probably related to dissemination and persistence of the infection, and the study of this response can give clues on new strategic treatment of Chagas disease. So that means that it's really complicated because of this feature of T-cruzi, that to infect almost any kind of cells and to induce different responses, completely different responses. The question was if there is a common pathway in the different models, not only in our experiments, but also in other experiments that are published in the literature, and we saw that this PI3 AKT pathway is a common response to T-cruzi infection. So different drugs targeting this pathway have been developed and employed in clinical trial evaluations. So this is there is an option to consider them as repurposed drugs for host-directed therapies in Chagas and other infection diseases. So that is a general conclusion. We have tried with some of those drugs like imatinib, glavoflavin, bisnagin, ICAR, rapamycin, and the results until now are negative. So we could not find good results by using this drug or combining this drug with becnidazole, that is the trypanoside. It is a drug used for Chagas disease. But at least this is the idea, and these are this table show is just to tell you that until now our results were negative using these inhibitors. Okay. And finally, I want to show that also the parasite readjusts its gene expression during the life cycle. What we analyzed was the transcriptome of epimastigotes, the insect stage, cell-derived trypomastigotes, and amastigotes. Yeah, and also we have found changes at the level of the transcriptional levels between the different stages of the parasite. Okay, here are some changes related to oxidative phosphorylation and Krebs cycle, in particular in some of these genes. Also changes, these are the colors: epimastigotes, trypomastigotes, and amastigotes. And these are, and here we normalized the read counts, and here are the different genes analyzed. So, for example, here those related to oxidative phosphorylation. Also, we have found in epimastigotes several genes that are overexpressed significantly with respect to the other stages in the pentose phosphate pathway, and also in steroid metabolism. That was not a discovery because we knew that epimastigotes are very active at the metabolic level. Just to show that we can use transcriptomic data to make inference about what is happening in different situations. After that, of course, we need to go to the validation of the data. Okay. Also, these are some examples in the glycolysis related genes. And these are some examples of nitrogen metabolism. For example, here, there is one of the amino acid transporter that is overexpressed in amastigotes, yeah, significantly, whereas another genes are overexpressed in epimastigotes. So this is just to show that some data can be used to go to the, for example, to the amino transport of amino acids and see some difference and what is and maybe this after functional validation, maybe this could be or should be a drug target, etc. So that is just to show some examples of how transcriptomic data can be used for metabolism. Hypothesis that what is happening in different situations, and after that we need to go to the metabolic studies directly. So this is an overview of what we have found in the mastigotes, trypomastigotes, and amastigotes. Yes, different pathways activated. For example, in amastigotes, anabolism, electron transport change, NADPH generation, nucleotide biosynthesis. In the case of amastigotes, well, some transmembrane transport that, for example, those related with some specific amino acid, and this changed also in trypomastigotes, signaling, and of course, surface remodeling, because it is known that the surface of the trypomastigotes must remodel to avoid to evade the immune system. Also, the parasite changes at the level of small RNAs. It is not well known what happened with the small RNAs patterns in parasites, and in cruzi there are different small RNAs when compared with humans. Just to show you that we have done, we have compared intracellular small RNA and extracellular vesicular small RNAs, and we see that there are different patterns in both situations. Yes, and also that there are some patterns at which we concentrate in these intracellular small RNAs. There are some small RNAs derived from tRNA, from snow RNA, and from those small nuclear RNAs that deserve to be studied more in depth. Okay, for example, here you can see that these small snow-derived small RNAs are different in the intracellular pool, are in the pool of small RNAs from extracellular vesicles. So, and finally, I want to show that in the literature, it's not so clear. In the literature, it is very difficult to conclude because the number of strains and the different strains, there are different strains that have been used. Yes, also the MOI is different between in the different works that make transcriptomics. Also the time of incubation, also the host cells, also the models in vivo or in vitro. So it is very difficult to get conclusions from those studies. There are several variables when you make transcriptomics. Where do you propagate the trypomastigos, the cell type, the strain of cruzi, the relation between parasites, the number of parasites and cells, if the studies are in vivo or in vitro, the interaction time, the analysis times, and the technology. Well, this is now most of the technology used are deep sequencing. So the perspectives we think that are to generate tools that allow the integration of different omics, this host pathogen crosstalk and target host with T. How to do it in networks? I mean, how to avoid this kind of tables and try to get a discussion about how to have common models between the different groups. So that is how to do it in networks. And I think that this workshop is a very nice initiative that goes in that direction. This is the group of our group. This is our funding. Okay, thank you very much. This is most of the people has participated in this work, and thank you very much.
Thank you very much, Carlos, for this very fantastic talk.